Hello and welcome to my sample lesson. My name is Rosalind and I'm a qualified science teacher. I can teach chemistry, physics and biology to GCSE or IGCSE, but I'm a specialist in A-level biology. Today's lesson is going to be aimed at those students studying A-level biology, and in particular if you are in your second year of biology, you will learn about something called dihybrid inheritance. So let's get started with that. So today, I would like you to revise and review what you know about monohybrid inheritance, and then you're going to learn how it's different to dihybrid inheritance. We're going to practice doing Punnett squares to be able to work out the phenotypic ratios for a dihybrid cross. So we'll start by thinking about the vocabulary you're going to need to know and to fully understand in order to understand dihybrid inheritance. So in a moment, I'd like you to pause the video and to write down as many words as you can think of to do with genetics and inheritance. Now, this might be something that you learnt last year in your first year of A-level, but if you can't quite remember that, think back to what you learnt in GCSE, some of the keywords that you used discussing inheritance. So I'm going to give you one to start off with, and that is DNA. So pause the video now and spend 3 or 4 minutes writing down as many words as you can think of to do with inheritance. So, how do you do? Which ones of these did you get? Did you get anything different from these words? So let's have a look at the keywords that I have written down. Allele, homologous chromosomes, locus, homozygous dominant, heterozygous, homozygous recessive, dominant, recessive, polygenic, phenotype, genotype, and F1 generation. Now you might have got some that aren't on that list, and that's fantastic. But what I'd like you to do now is pause the video and open up the attached worksheet. And what I'd like you to do is to match these keywords to the definition. It's really important that we are comfortable using keywords when discussing dihybrid inheritance. Biology is very, very vocabulary heavy. I always say it's like learning a new language, and when we're working on exams and answering exam questions, it's really important that we show the examiner that you understand what those keywords are and how to use them and getting them into your sentences. So the more comfortable with the vocabulary, the better your answers will become, because it will demonstrate what you truly understand. So video now and see if you can match up the keywords to the definitions there. So let's go through the keywords that we've got here and match them up to the definitions. So the first one we're going to do is allele. So allele, we can see, is a different version of a gene. So there are genes that code for our characteristics, and the alleles are the different versions of them. So for example, our hair colour. We have a gene that controls our hair colour, but there are many different versions of that gene. So for example, blonde hair or brown hair. Those are the different versions of the gene. The next one is homologous chromosomes. Homologous chromosomes are where you inherit 2 identical chromosomes. Chromosomes exist in pairs. You have 23 pairs in each of your diploid cells, and one of those chromosomes is inherited from one parent and the other from the other parent. They have genes in the same locus, however, as we've just discussed, they may be different alleles. The next word is locus. Locus refers to the location of that gene on the chromosome, so literally where it is placed on that chromosome. Next one is homozygous dominant, true breeding. What this means is if somebody or something inherits 2 dominant alleles for that particular gene, then they have a genotype which is known as homozygous dominant. Okay, the next keyword is heterozygous, and this genotype consists of 1 dominant and 1 recessive allele. Okay, so the next one is homozygous recessive. and this is where the genotype consists of 2 recessive alleles. An allele, what do we mean by that? Well, a dominant allele is always expressed in the phenotype if you only possess one for that particular gene. Whereas a recessive allele is masked by a dominant allele in somebody who is heterozygous for that gene, and therefore, in order to be expressing that characteristic for the recessive allele in the phenotype, the person needs to inherit 2 copies of this. Okay, polygenic is where there is more than one gene which contributes to a characteristic. Phenotype is literally the physical characteristics determined by the genotype. Now, it might not be something you see. For example, we can talk about your blood type, your actual blood type that you are. So, for example, if you have blood group A, that would be your phenotype. So it might not be something we can actually see, but it is your physical characteristics. Genotype is the allele combinations for a particular gene. So, for example, heterozygous for that particular gene is a genotype. But we give those genotypes letters, which we'll go through in a moment. And F1 generation stands for the first filial generation. This refers to the offspring produced when 2 parents are crossed. We can also think about the F2 generation. What do you think that might mean? We'll come back to that towards the end of the session today. So now that we've gone through the key vocabulary, hopefully that's started to warm your brain up towards inheritance. And we're now going to review what we mean by monohybrid inheritance, also known as monogenic crosses. When we're doing biology and we're thinking about these keywords, it's a really good idea to break the vocabulary down, to really try and think, what does that word actually mean? And if we think about mono, what do we think we mean by mono? We think about other words that might use a particular part of the word. Well, monogenic cross literally means the inheritance of a single gene. So we're going to have a think about how we put that into something called a Punnett square. Hopefully you can remember what they are from GCSE. So right now I'd like you to have a go at this one on your own first, just to try and really stretch and challenge yourself to see if you can remember it. So have a look at activity 2 on the worksheet now, and then pause the video to have a go. I'd like you to use the Punnett square that is on the worksheet to try to predict the outcomes of 2 true breeding pea plants when they are crossed. So one of those pea plants is going to have a homozygous dominant genotype, so it's going to be big R, and this codes for a plant that has round peas. And I'd like you to do the Punnett square where you're crossing that parent with its homozygous recessive. So that is where that parent has 2 recessive alleles, and their phenotype would be wrinkled peas. Once you've done that, have a think about what this test is. This is known as a test cross, but what is it and why are they used? So pause the video now and have a little think about that, and come back in a moment for the answers. So let's have a look at the answers to those questions. They are on the screen, you can have a look at them. I'll just explain. On the right hand side here, we can see what the test cross would look like in a Punnett square. We put the alleles for the parents in the boxes at the top and at the side, and then we bring the alleles together to predict the genotypes of those offspring. So we can see we've got the parent with the 2 capital letters, the 2 dominant alleles, big R at the top there, and then we've got the other parent down the side with the homozygous recessive alleles. We bring them together and we can see that we're predicting that 100% of their offspring will be heterozygous. What will their phenotype be? That's the really important bit. It's no good just giving the probability of the genotypes. We have to give what's called a phenotypic ratio. So 100% of these offspring would have round peas. Why? Because they have one dominant allele. And remember, the recessive allele is always masked if there is a dominant allele. So 100% of their offspring are going to have a heterozygous genotype. What if they produced 100 offspring and all of them had round peas? What would be the probability of the 101st offspring also having round peas? The answer, 100% every single time. So this is a test cross. What is it and why are they used? Well, a test cross is a way of working out what the actual genotype of the parent is. When we look at different organisms, we can see where the individual has the recessive phenotype. That's easy to spot. But when an individual has the dominant phenotype, how do we know if they're homozygous dominant or heterozygous? We can't actually, from the outside, see the alleles that they have in their genotype. So a test cross is done. You breed together the homozygous recessive parent with a parent that we want to work out, are they homozygous dominant or heterozygous? They breed together, and if 100% of the offspring have the dominant characteristic, and you'll probably have to do this over many different offspring, just to double check, then it's likely that that parent is what we call breeding. In other words, they're homozygous dominant. Okay, let's have a look at what we mean by dihybrid. So we just reviewed monohybrid. Remember, that was the inheritance of a single gene for a characteristic. But it's not really that realistic. In reality, thousands of genes go into making up the genotype of an individual, making up what they actually consist of and look like. So instead, we look at digenic cross experiments, in other words, the inheritance of two genes in order to control that particular characteristic. So I'd like you now to pause the video and have a look at activity 3 and see if you can fill in the spaces on the worksheet just to check what you think we mean by dihybrid inheritance. So let's go through those sentences then. So an individual's genotype will contain two alleles for each gene being inherited. Remember, in dihybrid inheritance, we're talking about two genes. So how many alleles in total will that be? That's right, 4. So we're going to be actually having lots of different combinations that we're going to see, many more combinations than we saw in monohybrid inheritance. Let's do the second sentence. Following meiosis, remember meiosis is a type of cell division which produces gametes. Following meiosis, each gamete will contain one allele for each gene that is being inherited. Now, this is a really important rule to remember because otherwise we're going to get really confused and we may end up putting more letters into our Punnett square than we actually need. So these are the basic reminders to help us with the next section. So let's go on to that next section. We are going to do a worked example together. So you'll probably want to write this down as we go through it. And then I'm going to give you an opportunity to do one by yourself to see if you can do it. So imagine that we are looking at the inheritance of two genes, A and B. So that's gene A and gene B that contribute to the characteristics of this organism. One parent has a gene that is heterozygous for both genes. A heterozygous means two different alleles, one dominant, one recessive. So this is what the genotype would look like for an individual that has a genotype that is heterozygous for both genes. So gene A, you can see is heterozygous. There is one dominant and one recessive. Remember to write a capital letter first. Then gene B also is heterozygous with the capital going first. We don't mix the A and the B at this point. We keep the E's to one side and the B's to the other. And always the capital letter goes first. I'll explain why that's important in a moment. So, if this individual produced gametes through meiosis, remember in meiosis there are 4 gametes produced. This genotype will produce 4 different gametes. Let's have a look at them. So, we bring A and B's together. Remember on the last slide, we said that there'd be 1 allele in each gamete for each different gene. So just bring the different combinations of the letters together. So big A, B, big A, little b, little a, b, little a, little b. So remember I said that we always write the capitals first, but you can see that that's we've got the little and the big B. That's just because we write them in alphabetical order. So it keeps it in the same format. Don't worry if this is a little confusing. I promise you, by the end, when you've done lots and lots of examples, you'll find it really, really easy. So these are the 4 different gametes that this individual could possibly produce. So now we're going to just check that you understand that, just that before we move on. So pause the video now and have a look at activity 4 worksheet and write down all the different possible gametes that you would get from these 2 genotypes. Now the first genotype I've given to you in letters, and the second one I've given to you in words, just to check that you understand that properly. If you can't remember what that keyword means, quickly look back to the keywords that we did at the start of the session, just to remind yourself. Remember, it's really important that you start using these words in everyday language just to get more confident with using them and reading them. So pause the video, go now, and then we'll go down and through the answers. So let's look at the different gametes that we got for this first one. So remember, we bring the 2 different letters together. So big A, big B. Big A, little b. And then for the homozygous recessive different, homozygous, remember that means that they're the same. Recessive means 2 recessive alleles for each one. Their genotype would be little a, little b, little b. And the different types of gamete that we could produce for that, just little a, little b. Now remember, I said that there are 4 gametes produced. However, we don't need to waste time writing them all down if they're identical. So for example, in question 2, all 4 gametes would be little a, little b. Do we need to write that down 4 times? No, because I want you to write down all of the different possible gametes. Well, on the screen are the different possible gametes. Even though big A, big B would be multiplied by 2, and then we get big A, little b multiplied by 2 as well. Okay, how did you do with that? Let's see about this next activity. So this is the worked example. We're going to go through really, really carefully, and again, write it down as we go in a series of steps so that you can look back at it when you have a go at one on your own. Plant heterozygous round fruits and unbranched flower genes was crossed with a plant with elongated fruits and branched flowers. Calculate the phenotypic ratio of the offspring. Now, elongated just means that they're slightly longer as opposed to round, okay? So let's have a look at how we do this. Well, the first thing we've got to do is we have to work out the genotypes of the parents, because once we've done that, we can work out the gametes. So the genotypes of these parents, the big R, little r, big B, little b. Now, in some examples in exam questions, they give you the letters, but in some they don't. So you just have to come up with the letters yourself. My advice is that you make them equal to that characteristic you're thinking about, otherwise you might get a bit confused. And also make sure that when you're writing down the capital and the lowercase letter, that they are distinctly different to each other. So they don't look too similar. You don't want the examiner trying to work out, oh, is that capital, is it a lowercase? Make it really, really clear, write it down. So big R, little r, big B, little b. So this is a plant that's heterozygous for round fruits and unbranched flowers. So we can see the capital R gives round fruits, that's the dominant characteristic, and the capital B gives unbranched flowers. And it's been crossed with a plant with elongated fruits and branched flowers. So in this case, the elongated fruits are going to be a recessive characteristic. So that's caused by the two little r's there. And the branched flowers is caused by recessive alleles as well. And so we've got the two little b's. So those are the parent genotypes. Once we can then work out the gametes. So I'm going to write down all the different gametes here, just as we did before. Remember, we don't need to write all four of the little r, little b down because that's just wasting time. We know it's multiplied by 4. Okay, so once we've done that, we're then going to create our Punnett square and we're going to put them together in the Punnett square. Okay, so write the gametes from one of the parents at the top and the gametes from the parents down the side, just like we did in the monogenic cross. Only this time, remember, there are going to be more boxes because we've got more gametes. So we put them together in the Punnett square here, and remember we've got only one different type of gamete here, so we don't need to write it all down. Sometimes these Punnett squares become really, really big because there are 4 different gametes along the top and 4 different gametes along the bottom. So sometimes we need up to 16 spaces for the offspring. In this case, we only need 4. Okay. Let's bring them together just like we did in monohybrid inheritance. And remember to write the capital letter first. This will really help us to spot the dominant characteristics when we're writing down the phenotypes of offspring. So, okay. Now the next thing that we're going to do is we're going to write down the phenotypes. Remember I said before that when we're doing these Punnett squares, we can't just write down the types. We have to give what's called a phenotypic ratio. So that means the different phenotypes and how many of each that we have, so the portion of each type. So here we've got round, unbranched. That's what this offspring would look like. Round because it's one round dominant allele, and unbranched because it's got one unbranched allele there that would be. What do you think the next one would be? Have a little think. I know you got it right. It's branched. This one, elongated and unbranched, and this one, elongated and branched. So now we have the ratio, portion of each one. So the ratio, as we can see, we can just count them. We've got one round unbranched, one round branched, one elongated unbranched, and one elongated branched. So the ratio is 1 to 1 to 1. We're always looking for 4 numbers here, okay? And all we do is we count them and we do the ratio. So hopefully you found fairly good. If you didn't, I would suggest you pause it, go back through the example again, and then we're going to move on to the next one. So I'd like you to pause the video now and have a look at activity 5 on the worksheet. Now this is a slightly different example, a slightly different genotype to start you off with, but you do it in exactly the same way, the same steps. Work out the parent types, work out the gametes, put them together in a Punnett square, work out the phenotypic ratio. So video now and have a little go at that. Okay, the answers to activity 5. So at the top you can see we've got the parent genotypes, and then written down the different gametes, and then we've put the gametes for one parent along the top, the different gametes for the other parent along the bottom, and then we've counted up how many we've got. So our ratio, as we can see, down our phenotypes, we've got 3 disc-shaped and white, 3 disc-shaped and yellow, 1 sphere-shaped and white, 1 sphere-shaped and yellow. So our ratio is 3 to 3 to 1 to 1. It's really important when we're doing questions on dihybrid inheritance that we use the words in the question to write down the phenotype. We mustn't just change we mustn't change what the characteristics are of that particular organism. So just use the words from the question to write the phenotype. Okay, let's go at this one then. This is slightly different again. This is where there are 2 plants that are heterozygous for both genes. And this is a really important one. I'll explain why in a moment. But this is going to be a much bigger cross. So you're going to want to leave a little bit more room on your page for your Punnett square and see work out the phenotypic ratio of the possible offspring for this plant. Pause the video, have a go, and we'll go through the answers in a minute. Okay, here are the answers. Hopefully this has got 2, but please don't worry if you didn't, because we can practice this lots and lots. There's so many different examples we can do. So this is the one that I was talking about, where you've got to leave 16 spaces because of the 4 gametes that we've got for each parent. And we bring them together into the boxes in exactly the same way. So writing the capital letters first. Now this is really important because when we get a more complicated cross like this, we need to sit and write down all of the different phenotypes that we're seeing. And if we've written the capital letter first, it's easy to just spot them. So when I'm going through and checking, I look first for those with the capital D and the capital W, because they've got the dominant characteristics for both of these different genes. So you can just scan as you go through, just scan those. You can see I'm scanning for capital D's and W's and counting them. So if we go through and count all of these different ones, this is what we should have got, a ratio of 9 to 3 to 3 to 1. I want you to say that again. 9 to 3 to 3 to 1. 9 to 3 to 3 to 1. This is always the expected ratio when 2 heterozygotes for 2 genes exist. So in any dihybrid cross where the 2 parents are heterozygous, remember that means that they have a dominant and a recessive allele present for both genes, the ratio is always 9 to 3 to 3 to 1. That can save you time in an exam question where perhaps you don't need to draw down the entire square. If there's not that many marks available, you probably don't need to. You may just need to give the ratio 9 to 3 to 3 to 4. Or if you do need to draw the Punnett square, it's self-checking. You already know what the answer is. 9 to 3 to 3 to 1. Got to say it over and over and get it into your head. So this is also really important because the next lesson, the one after this, we would go on to look at carrying out a statistical test called chi-squared. And this is to determine if there is a significant difference in real life between what we expect in the Punnett square, the 9 to 3 to 3 to 1, and actually see in real life. Is there any significant difference? And if there is, what are the reasons behind that? So that we would go on to look at next time as a statistical test. And I'll leave you with what we have learned today. So now you feel a little bit more comfortable with the vocabulary that we need to use for genetics and for inheritance, and in particular dihybrid inheritance. And we've revised monohybrid inheritance, which is the lesson that you will have learned prior to this. And you can now calculate phenotypic ratios for dihybrid inheritance. And you should remember that if the two parents are heterozygous for both genes, then the expected ratio is always 9:3:1. And little task that you can have a go at now before what would be the next lesson, and that's another little example for you to have a go at. And in the next lesson we would then go through that question together to just check that you've been able to do it. Okay, thank you very much for watching, and I'll see you soon. Bye bye.